Okay, welcome to video 3 in our course F1 on flow cytometry data analysis. Now we're going to look at transit transformation, batch effects, and normalization. So in this instance, we're going to look at how we transform our data, which is notionally linear, into a log high-risk scale, or most typically that's called a by an exponential scale or by an exponential transformation, although slightly different transformations do exist outside of that. So what we have to bear in mind with our flow cytometry data is that due to the spreading error and fluorescence baseline subtraction approaches, we will end up with some negative values, even though negative fluorescence is kind of not possible in theory. In practice, we actually have some negative values. And you see these on the plots here. So what you note with these scales is that the 0 here is along these two axes. And you can see that there are a substantial number of cell points that are on the negative scale, and that can be for both components here. Consequently, representing the true components of variation in our data is something that's of interest. And that would mean if our key components of variation here are say across this area, maybe up to this point, and the same on this range for the other axis, we don't necessarily want to focus in on this area. So we're effectively transforming the visualization of our data and focusing it on a different way, in a different way. And the same way we have colors up here, we have a kind of contour map of the same thing down here. And the same applies on the right. So this is a simple log transformation that you're familiar with, going from 0.01 all the way up to 10 to the 5 here and 10 to the 5 here, and same down here. But you can see here, due to this, these negative values, you get a lot of values scattered down here, which are not really uniform clusters. So that's why the by exponential approach is taken to allow these negative values to exist, and so that they don't cluster together as a single group. And you see here we have a lot of values actually associated with being negative for both items on this plot, as well as ones that are clearly positive for this x-axis item, and those on the y-axis that are also positive for that item only, and few really that are positive for both in this kind of quadrant up here. So if we have this plot, we can see the same sort of pattern of the 4 major clusters. It's just that a lot of space is taken up by the ones down here that are have very small levels. So that means it's just a different way of representing our data, and therefore we should be conscious of what's that actually doing to our data. The details of the transformation approach depend on what data we're looking at. So there's no perfect way, but it's just a different way of viewing our data. So some platforms will need different parameters called cofactors for transformation because they're detection systems, such as looking at photons of light versus heavy metals. And we think about instances where we have a clear cut difference between two cell types, where you can see this is kind of almost centered on just over 0. And therefore we have a clear distinction between one with this cell marker, CD3 positive cell marker, one without. So if you look at example data, this is from a mouse spleen analyzed by fluorescent flow-based approaches, spectral data, or mass spectrometry. So that's the top, middle, and bottom. You can see here for the top one, we have certain cofactor parameters for each of these that differ across the different experiments. They're all not the same, even though the variables labels on each of the axes are the same. And what you can see at the top right is the clear separation of these one into 4 major groups, well, three major groups with really nothing here on the right. But you can see there's quite a lot of samples that are intermediate here and don't clearly separate. And we kind of get the same pattern here in the second image, and a comparable pattern here in the bottom image. But note, for each of them, the cofactors differ widely. And that leads to one of the major issues with this transformation of flow cytometry data, is that we use the wrong cofactors, and the wrong cutoffs can make things more difficult for us. And that can lead to other things that we might have one single population in reality. We might get 2 peaks symmetrically aligned with around the 0 on the x-axis, and this gets interpreted as 2 distinct populations, which is not actually true. And I guess the principle behind by exponential plotting is that the cells within the range of autofluorescence, but that's in the bottom left here. If you look back at these images, the ones down here, are not of interest here because they don't show anything interesting. So an example of bad plots with the wrong settings where the split the negative population to two visually distinct groups, where the red area shows the true population of CD3 positive cells. You see that the red area is shown in here, but we got this kind of spreading of the cells out across from the negative to the positive here due to this kind of the series of correction procedures that we implemented. So it's for a high cofactor that kind of gradually decreases. If we drop down to a cofactor of 500, we still see the same error is built in there. Cofactor of 50, that's lower again. A cofactor 5, we get better separation. So you can see the CD3 positive cells on the x-axis, but you do still have separation on the y-axis. So that's an example of bad plots over here for when you use a very high cofactor. So this can mangle our results, and therefore we have to be careful about false mergers or splits among our populations, and using negative or positive controls are instructive in that point. We could also look at the same data and look at TSNE, T-stochastic neighborhood embedding, and how that's really ineffective here because it kind of mixes the clusters together in an unreliable fashion. So if we use the same height cofactor here of 5,000, what we see for the two TSNE dimensions is that we have certain regions here where we have populations clustered together that aren't necessarily present here at lower cofactors. And if you look at the black and white arrows, we can see the separation of certain cell populations into two different clusters, even though there are none really present. So that's kind of shown by these different arrows where we have these clusters that aren't really valid ones. So with TSNE, because it represents many dimensions on a single plot, we should interpret it with caution. So batch effects are another key consideration in this data transformation process, and that can be caused by variation across time, the instrument type, the reagents being used, the samples, as whether there's is there randomization? Is there blinding to make sure that the samples are not correlated with particular instruments or particular days on which the experiment is run? To get around this, sometimes manual gating is used, but that can also lead to batch effects too. So it's possible to reduce these batch effects computationally, but also we can reduce it by adding a control sample to each batch so that at least we can correct for those effects that are identifiable across different batches. I remember that our underlying assumption about all the experimental data is that the errors are random, and therefore we can distinguish them from batch effects that are systematic. But distinguishing batch effects associated with an experimental machine, for example, distinct from biological effects can also be tricky unless we've designed the experiment in the right way, such that blocking is used to allow for block-based allocation of different samples such that they're represented across the different batches so that we can correct for that property. One simplistic approach is to align the different matches based on their minimum, median, and maximum values. But there are nonlinear approaches as well that are probably superior. One is called Cytobatch adjust, Cytonorm, and SwiftReg. And in principle, what they're trying to do is take this distribution like this, where maybe there's two underlying distributions, and kind of consider if they're occurring because there's a shift peak due to different sizes, or is there a different intensity present? So that's from SwiftReg, which details that approach in more detail. So that concludes that part on looking at batch effects, data transformation, normalization that we'll expand upon at the labs. Please see the next video on data clustering.